Well, thank you, Christina, for that introduction, and thank you to Professor Cittadini for twisting my arm to come to this beautiful city of Palermo. I'm sorry I can't spend more time here and have a look around this historic island. So I was given the title of Guardando dentro l'embrione. I don't speak Italian, as you can tell. Looking inside the embryo. And my first thought was actually this image from one of my students, Caterina Chatimiletiu, who's now working at the University in Thessaloniki, back in Greece, which is a scanning confocal picture in which the tubulin has been stained in green, and blue shows the DNA of the nuclei. And I'll come back to the significance of that later in the talk. But basically what I'm going to do today, I'm associated with pre-implantation genetic testing, but I'm going to demonstrate that these tools can also be used to answer questions about the development of the early embryo. And just as fish, for example, highlighted the mosaicism that we have at cleavage stages, then the new techniques of SNP genotyping and carrier mapping, which allow us, if you like, to paint each chromosome by these molecular genetic markers, can allow us to work out what's happening within the embryo. Okay, so first of all, the historical context. I was actually around in 1978 when Louise Brown was born, and actually I helped, Bob Edwards came back to Cambridge, to the Marshall Lab, and there was a celebration there with Jean Purdy, and in fact, Steptoe couldn't join us at that time for obvious reasons. And some years ago, I was actually a student of one of Bob Edwards' first students, Martin Johnson, and he arranged a 40 years of IVF celebration in 2009, which related to the key publication in 1969 by Bob on human egg maturation in vitro. It was a very nice multidisciplinary meeting, and in it they actually showed this photograph. Now, this photograph here in the lab at the celebration party, which is where I was, probably taking this photograph. And you can see here Barbara Rankin, which is Bob Edwards' long-term secretary, holding a cartoon here of the three of them, which no longer exists. Unfortunately, Bob lost it, probably deliberately. It wasn't a very good cartoon, but it was a cartoon of the three of them, Patrick Steptoe, Bob Edwards, and Jean Purdy, holding a test tube here. And that was drawn by me in 1978. So that's my memory of Louise Brown's birth. So, looking inside the embryo. I've recently been to a meeting in which a new company has been set up to go back and look at the possibility of flushing embryos following normal conception in vivo. So I'd like to share those images with you because I think the clinical embryologists in the audience will be fascinated to see what embryos recovered from the uterus actually look like. We've only previously had the Carnegie images and so on. And I think this is really an eye-opener, and I've got permission to show some of those images. So we're going to look inside the embryo, and I'm going to show what the embryo looks like when you peel off the outer layer, the zona pellucida, which surrounds the embryo. Some beautiful scanning electron microscope pictures taken in the early 1990s. And then show you the results, again, that go back quite a long way, in which I differentially label the nuclei of the outer trophectoderm cells that will form the placenta after implantation and the group of inner cell mast cells. And that allowed us in the mouse actually to look very carefully at the increase in the numbers of trophectoderm and inner cell mast cells, which are the two lineages, the embryonic lineage and the extraembryonic lineage, and show that there are significant differences and actually quite significant cell death, which is actually quite now interesting in relation to doing genetic testing in spent culture medium. And finally, I'm going to show you then how this SNP genotyping technique can be used to paint the chromosomes metaphorically and actually show how they distribute themselves in normal and arresting embryos. And this has really been a revelation, to me at least, that in fact the abnormal segregation of chromosomes is probably responsible for the lack of development of embryos to the blastocyst stage. So I've wondered for 30, perhaps 40 years, why only 50% on average of embryos go through to the blastocyst stage. We're now beginning to get some answers. Okay, so alla cacciare la cintura. Fasten your seatbelts. Okay, so firstly, the in vivo conceived human embryos. And I think this is fascinating. We have here an unfertilized egg. So this is following insemination in utero and flushing on day 5 and day 6. And we see some really quite abnormal embryos, including this embryo here, which we often see following IVF, as well as some very nice examples of blastocysts. So I think we can conclude that actually embryo arrest probably also occurs in vivo, but this is going to be a very interesting area of research. We'll now, for the first time, be able to compare in vivo embryos with in vitro embryos. And I can tell you that, because it was presented at the meeting, that actually they've looked for chromosomal aneuploidies and find a high degree of aneuploidy in these embryos as well, despite the fact that these are young women that are involved in the trial, which actually is in Mexico, but they have IRB approval from the US. And finally, just to show that along with the abnormals, they also have some very beautiful blastocysts, and this is a beautiful hatching blastocyst with a very large inner cell mass. So this is beautiful. So then to peel off the outer layer, we can look by scanning electron microscopy from day 1 all the way through to day 6 here. And in fact, the zona is still present on this embryo, and it's actually hatching from the zona. And you can see the features of the outer surface of the embryo. It's covered by microvilli, and here we have the first and second polar bodies. Then on subsequent days, the embryo cleaves, of course, and then between days 3 and 4, it compacts. And so what we see, actually, you may be able to see that when, during shrinkage, the cells peel back, you can see microbilus-free areas. So the microvilli become localized on the surface of each of the cell of the morula. So then as this then begins to form tight junctions, it can accumulate fluid and form a blastocyst, which then eventually hatches, and this is a fully hatched blastocyst here. So these are beautiful pictures. And I know that Yorgos has retired to the hills above Athens and actually has a scanning electron microscope in his basement. And I think when I retire, I shall take a fluorescence microscope and put it in my cellar. Okay, so I'm going to go back to some very early studies in the mid-80s that I did on mouse embryos using this technique of labeling the outer trophectoderm nuclei in red or pink here, and the inner cell mass nuclei here in blue. So this isn't related to any specific gene expression. We can now do this with probes, for example, to nodal and other molecular markers, of course, but this was purely based on the physical separation of the trophectoderm and the inner cell mass, and I'll show more examples of that. Obviously, the inner cell mass is the embryonic lineage that gives rise to the baby and minor components to some of the extraembryonic tissues, and the outer trophectoderm is responsible for implantation. So by using differential labeling, you have the advantage that you can look at the numbers of cells in each individual embryo instead of separating them and then doing averages. And this was my experimental model at that time, the mouse. And you can see you get beautiful development, absolutely perfect each and every time, which is very different from the human embryo. That's not quite true, but Okay, so when I moved across to human embryology in 1986 to join Robert Winston, now Lord Professor Robert Winston, I had various objectives, and one of them was really to really duplicate these studies we're still in the mouse. I apologize. So I'll just show you the results that we got from the mouse. So in the mouse, we actually recovered embryos in vivo, developing embryos, I think every 3 hours from day 3 to day 4, until the point at which the embryo was beginning to implant in the uterus. So we could follow very carefully in vivo how the population of trophectoderm and inner cell mass cells increased. And when we did that, and this is just an example, more examples of the technique. So we used the technique of immunosurgery to specifically lyse the trophectoderm cells and label them in red with propidium iodide. And then we came back and fixed the embryo and labeled with DAPI so that we had differential labeling of the nuclei. But we could still distinguish, by changing the excitation filters, we can look specifically at the trophectoderm population or the inner cell mass population. And what this shows is that we can find cells in metaphase, so we can work out the mitotic index in the two lineages. And we could also see cell death, and you'll see examples I can't see exactly from this angle. You can see cell death in the inner cell mass. And in the mouse, it was very striking that at a time when if you isolate the inner cell mass, it can still regenerate trophectoderm, there was a loss of cells through apoptosis. Coincidence with the loss of that ability to reform a layer of trophectoderm, telling you something about how the development of the lineages becomes fixed within the embryo. If you look at the curves, the population curves, then in fact you had doublings in the early stages, and then it gradually tails off in total numbers, and the trophectoderm is by far the majority of cells in the embryo at this stage, but the inner cell mass failed to double, and that's because of this wave of apoptosis. So we then, with one of my other students, now a professor in her own right, at Hammersmith, still based at Hammersmith Hospital, we began to look at the human blastocyst, and I think, again, looking back, I think it was Simon mentioned the fact that we were culturing embryos in a different way in 1989, and these embryos were cultured in Earl's balanced salt solution, which was made up by the embryologists, and heat-inactivated maternal serum. And so the maternal serum was different for each patient. We had very good development of blastocyst, and indeed, if you maintained the culture beyond day 7, you had very good development. I think current media are actually very poor at maintaining development beyond, I think, about day 6. So all of these results that I'm showing you here relate to that medium, which is quite different. I should also say that Robert Winston had already tried to transfer embryos at the blastocyst stage, and after 20 transfers with no pregnancy, he stopped. And so we continued to do cleavage stage transfer, even though we could develop embryos to the blastocyst stage. So you can see here the numbers that we found in embryos on day 5, 6, and 7. Clearly, these were only small numbers of blastocysts, but remember that in comparing the trophectoderm and the inner cell mass cells, you're comparing the two populations in the same embryo. So it's quite a powerful analysis. And we also looked at the mitotic index and the dead cell index. And what we appeared to see was some synchrony still remaining from cleavage, so that we got a doubling in the trophectoderm between days 5 and days 6 and 7, but the inner cell mass and the inner cell mass only began to show an increase on day 7. so that they're synchronous but out of sync. And this was also shown in the mouse at early stages. The significance of the dead cells, the fragmenting cells, which are typical of apoptotic nuclei, is, of course, now that we know that there is DNA in the spent culture medium, and there's a lot of excitement about recovering this material. And actually, and I see Carlos Simon in the audience, has just published some evidence on this. There's some concordance then with the genetics of the embryo, but some controversy about how well that actually reflects. But I think there's certainly evidence that there is some cell death. But remember, this is in that crude culture medium. So this is the real puzzle that I'll come to in the last part of my talk, which is that this was a very early culture medium, but even later culture, the development of later culture media that have led, I think, the way to much improved IVF success rates, nevertheless have not improved the proportion of embryos reaching the blastocyst stage. So why is it that 50% of embryos don't develop, which is so different from the mouse, where you can get upwards of 90% of embryos to develop? So these are just the pictures. The differential labeling is not as striking, but it is beautiful to look at in the microscope when you can focus through the embryo. But sadly, we didn't have 3D reconstruction at that time, because some people at PGDIS in Bangkok recently showed some very nice fluorescent images in which you can actually now rotate the embryo and show the 3 dimensions. So you could actually then pick up where these clusters of dead cells were and ask questions about, is it a subpopulation of cells in the inner cell mass, or is it spread throughout these 2 populations? So these studies led on to a lot of studies using immunofluorescence and just labeling the nuclei here. And here you see 2 examples. So in the early 90s, we found a lot of abnormalities of nuclei, and these are just 2 very striking examples. In this case, both of these cells are binucleate, and you have multiple nuclei in this and here. And so you have multiple nuclei in some blastomeres, and you have 2 normal-sized nuclei in some blastomeres, which actually, when you look by fish at least, both have a normal complement of cells. And we also found by fish a lot of tetraploids. So this led to the idea that these nuclear abnormalities could secondarily cause mosaicism. It's also interesting to note for the embryologists in the audience that actually the nuclei get progressive. It's not only the cells that get progressively smaller during cleavage, but so do the nuclei. And this is because the chromatin components are changing in this early period of development. And on the other side, you see another example, which when I take the red label off, you can see multiple nuclei in these cells. And that's obviously why these embryos are arresting. Okay, so it was in 2005 that Katerina Chatzimarletiu, working in Leeds at that time, we got some probes to tubulin and pericentrin and some other components of the spindle. And we had this paper in Human Reproduction with a very nice picture of a labeled embryo on the cover. And so Katerina showed that with spare embryos that we were allowed, we had patient consent to use for these studies, had really a striking number of spindle abnormalities. So if you label the tubulin in green and the DNA of the chromosomes in this case, you can show that you have really quite strange spindle structures, in this case tetrapolar spindle. And that, of course, will affect the way the chromosomes segregate in the early embryo. So the early embryo then has a constellation, if you like, of abnormalities that can lead to genetic issues of various kinds. This is just another example And in this case, if you switch the tubulin staining off, you can see chromosomes lagging that have not congressed onto the bipolar spindle. It's obviously a very poor morphology spindle. And then back to the beautiful image of the blastocyst, and you may pick out the various mitoses, the cells in metaphase here. That was the rest of my lecture. I don't have to give that now. So firstly, there's a tripolar spindle. And that was, so it was known that abnormally fertilized embryos could divide not into 2 cells, a normal bipolar division, but could divide into 3 cells, giving you a quasi-diploid embryo. So it could resolve itself. So after abnormal fertilization, the triploid embryo could divide into 3 cells. But here we demonstrated that actually these tripolar divisions were still occurring in the trophectoderm. And this is probably one of the reasons when we take a trophectoderm biopsy of trophectoderm cells that we find chromosomal mosaicism in this lineage. And this is still an ongoing and very hot topic in the genetic testing community. But then you see other bipolar divisions and other abnormalities. So what I'd like to talk about in the last 5 minutes is work that we've done much more recently using SNP genotyping. SNPs, as you were already introduced this morning, are genetic markers, changes in nucleotides that occur periodically in the DNA of all of us. And at that particular point, we have a different nucleotide. Now, at one end of the spectrum, that could be a mutation in the coding region of a gene. But at the other end of the spectrum, they have no known function, but they are very useful to label that chromosome in that individual because there is a nucleotide difference. And so we have microarrays that allow us to sequence quickly and cheaply hundreds of thousands of these SNP markers spread across the genome, across each and every chromosome. So this, if you like, I don't want to go into too much detail, is a molecular genetic painting, which once we've processed the data, we need to look at the parents and how it's being, actually allows us, as we demonstrate in this paper, to do 45 chromosome fingerprinting. So we can fingerprint each of the individual chromosomes, except the Y chromosome. So we have a fingerprint for each of the 45 chromosomes in the embryo. 23, obviously, inherited from the mother, 23 from the father. Okay, and what this has shown is that tripolar divisions in early development lead to the dispersal of the chromosomes and the partitioning of the genome. And for that reason, once the cells have developed to approximately the 8 to 16 cell stage and are dependent on the activation of the zygotic genome, so they're not dependent on the egg, on mRNAs and other factors in the egg any longer, but must start expressing the embryonic genome, then they stop. And they're either excluded if there's only a minority of those cells, or in fact, it blocks development of the blastocyst completely. So tripolar divisions have been seen now in many different, and have been reported in many different reports. Carmen Rubio and Carlos Simon have reported on this. And I just show here some examples from this particular study. And on the top, you probably won't be able to see all the details, but if anybody's interested, I can either send the paper or we can look on my laptop. You can see here normal development, cleavage to 2-cell, and so on, through to the blastocyst stage, and this one is actually beginning to hatch into the ceiling. It's implanting in the ceiling. In this one, there's actually an abnormal division at about this stage in the third cleavage, and those cells we can follow and are actively excluded from the resulting blastocyst. So you commonly see cells excluded from the blast cyst. All right? And what we've done is to recover those and ask the question, are they chromosomally normal, those cells, or not? And you'll see the results. And then in this situation, and this is the example I will show, there's really, it's quite remarkable. You have normal first cleavage to the 2-cell stage, then you have it cleaves to the 4-cell stage, but one blastomere divides from 1 cell to 3, and then the other blastomere also divides into 3. So we have 6 cells suddenly, but they have arisen through 2 tripolar mitotic events. Okay, so how are we going to follow the dance of the chromosomes? Well, what we do is we run these SNP markers on the parents and perhaps an existing child or another embryo, and we say what we set is phase. So we now have blue and red chromosomes in the father. This is chromosomes 1 to 6. We're just showing you for example. And we define the mother's chromosomes, the markers on the mother's chromosomes, as green and yellow. And the reference embryo or sibling is here. And of course, that's by definition. So we set the markers up. Now when we look in the embryo, we see Mendel's genetics, because we now have recombination during meiosis in the formation of the egg and also in the formation of the sperm cell. So we have a unique pattern of recombination across. So this recombination pattern is different from this recombination pattern in these 2 embryos. So we have a chromosome-specific fingerprint, and we can do that for all 45 chromosomes. So carrier mapping is, even if, I think the lowest we've seen are 8 chromosomes in 1 cell. We can still pick up those 8 chromosomes and show that we have the exact same pattern that we have in other cells in that embryo. So this is the explanation of how it's laid out. So this is the paternal chromosome here, this is the maternal chromosome. We're now in the embryo cell. We've got a pattern of recombination, which is different between embryo 1 and embryo 2. For the paternal chromosome and the maternal chromosome, the gap is the centromere, and we have the P arm and the long arm here. And so that's really essentially what we're looking at. So what we did, we took the cells from the arresting embryos and the cells that were excluded at the blastocyst stage and asked the question, what is the chromosomal complement? And I was absolutely shocked at the result. And I just show you an example here. So these are the results for chromosomes 1 to 6 for a number of cells from this embryo that I showed you on the initial time-lapse panel. And this is one cell, this is another cell, and so on. This cell has this complement, and this cell has exactly the same complement. So it's missing both the paternal and the maternal chromosome for chromosome 1 in all 3 cells. It only has the paternal chromosome for chromosome 2, has the normal pair of chromosomes for chromosome 3, and so on. But the pattern is identical for 3 different cells. And the only explanation for that is that they are related and that these are clones of cells. So this is the first time we've been able to demonstrate that tripolar mitosis causes an abnormal segregation of chromosomes, but then the embryo continues to develop, continues to cleave for perhaps 1 or 2 or 3 further divisions, but it is then dependent on the embryonic genome. And of course, you can imagine that chromosome 1 is one of the largest chromosomes. And it's not present. It's completely nullosomic in these cells. So it is pretty obvious that its development is certainly going to be compromised at some stage. And what appears to be happening is that it fails to compact and it fails to incorporate then into the blastocyst. So that if this happens later in cleavage to only 1 or 2 cells, then only a few cells are excluded, and you can still get the other cells, which have the normal chromosomes, forming a blastocyst. So if I compile, if you like, the karyotype, so this is just, I'm sorry, I've gone over. Do I have another 2 or 3 minutes? Thank you. So this is just a diagram of 2 pairs of chromosomes segregating on a normal bipolar spindle. And obviously you end up in the daughter cells, as you would hope, with 23 pairs of chromosomes, 2 of which are shown here. If you have a tripolar mitosis, you have 3 spindle axes, and if they randomly, what we say, congress to each of the 3 spindles, then what happens is in the 3 daughter cells, you have a reduction in the number of chromosomes. And for example, in this cell here, you don't have either chromosome for the larger chromosome. So what happens then is that you have 46 chromosomes, you've replicated them to 92, you then take the 92 chromosomes and you divide them up randomly to 3 poles. So instead of 46 chromosomes, you only have 30 chromosomes on average. So you've lost a very significant part of the genome. And when we compiled this data, so this is the embryo which we saw by time lapse had 2 tripolar mitoses at the 2-cell stage. And what did we find? We found, if you look, we found 6 clones of cells with identical or complementary chromosome complements. So we can, in fact, recapitulate. So the most important thing I want to point out is that we also did the polar body analysis in these embryos, and there was no sign of any paternal meiotic error and no maternal meiotic error. So this embryo started life as a euploid embryo, divided into 2 cells, and then divided to 6 cells. But, so then you have these clones of cells, and in fact you can almost see this gap here, you see this gap here is down here. So the more you look, the more complementarity you see. And these have identical chromosome patterns. And remember, this is based on that fingerprint that we have, the recombination fingerprint. And then in the panel, again, this will be too small for you to see, but in the final column is the percentage of nullosemy, the percentage of the genome that's missing. And so you can have from about 4% to 50% of the genome missing from these cells. So in our study, we looked at over 100 cells from arrested embryos and from those excluded from the blastocyst, and over 90% of them did not have the complete genome. So we think this finally, at least for, I think Carlos Simon's group estimates about 25% of embryos that undergo these abnormal divisions at different stages. This is an explanation for the lack of development of blastocyst. And I believe that also asynchrony in other cell cycle parameters probably accounts for the other arresting embryos. So this is my concluding slide. Tripolar mitosis is relatively common given our current culture conditions. So I think the interesting thing, so what I've demonstrated is that a euploid embryo can end up as highly chromosomally abnormal, preventing its development. The dispersal of the replicated chromosomes in a tripolar mitosis leads to subdiploid chromosome sets. And if this occurs early in cleavage, you get clonal development and can show that you have virtually identical chromosome sets. and this partitioning of the genome then blocks further development. Okay, thank you very much.